Hi guys, today we're going to be learning about the tryptophan operon. So what does the tryptophan operon do? It basically turns on and turns off the genes that allow for the transcription of enzymes in E. coli that synthesize tryptophan. Okay, so in ways the tryptophan operon is basically the opposite, its functionality is the opposite of that of the Lac Operon, which we learned about in the previous episode. But before we get into the differences in functionality, I'd first like to show you the structural differences. Okay, so this regulator area that produces the repressor is basically known as R. Instead of I, we just call it R for regulator, and that is what produces the repressor. So similar to the Lac Operon, this regulator area is not a part of the actual operon, because an operon is a collection of genes that have a single promoter and a single terminator. But this is still important to the functionality of the entire operon as a whole. That's why we include it in the diagram. Now another difference is that the operator is actually located within the promoter. Okay, and then instead of having 3 structural genes like we do in the Lac Operon, here we have 5 structural genes. And those 5 structural genes are those structural genes are E, D, C, B, A. Okay, but in the Lac Operon, we saw that 3 enzymes were being synthesized by the transcription of these genes, and there are 3 enzymes that were synthesized by the transcription of the 5 structural genes in Tryptophan Operon. Okay, so now what are the differences in functionality? So as we saw in the previous video, in the Lac Operon, if there's a concentration of lactose, then you would need these genes to be transcribed so that lactose metabolism could occur. And then it would need to be turned off if there was no lactose. Well, the Tryptophan Operon is the exact opposite. If there was a concentration of tryptophan, then you would not need these genes to be transcribed. Why? Well, because the Tryptophan Operon is linked to the synthesis of tryptophan. If you already have tryptophan, you don't need to transcribe the genes that allow for more synthesis of tryptophan. However, if there was no tryptophan, then you would need these genes to be transcribed so that they could produce the enzymes that would synthesize tryptophan. So how does this controlling work? So basically this R gene, which is similar to the I gene because it is what codes for the repressor. So it would code for a repressor. However, the major difference from the Lac Operon is that this repressor would be inactive, meaning that upon immediately being transcribed, it won't be able to bind to the operator region. So here we could see that there is no tryptophan. That means transcription of these genes needs to occur so that the tryptophan can actually be synthesized. And since this repressor cannot bind, these genes will actually be transcribed. But now pretend these genes are transcribed a lot of times and then there is tryptophan available. That means that you no longer need these genes to be transcribed to synthesize more tryptophan. So what would happen then? Basically, this is a repressible system. The TRP, or the tryptophan, works as a co-repressor. What it does is that it binds to this repressor, and that actually activates the repressor. It makes the originally inactive repressor active. And what happens after that is that the repressor actually goes and is able to bind to the operator. So as you can see, there's tryptophan available, so the tryptophan can bind to the repressor. And because of that, transcription of these genes will be stopped because there is already a sufficient amount of tryptophan. Now there is another aspect of controlling this operon that comes in, which is attenuation, and we will discuss that in the next video.